There are two basic types of chemistry. Organic chemistry is the chemistry of organic molecules. Organic molecules will contain both carbon and hydrogen. Organic molecules are held together by covalent bonds, and they tend to be large molecules, which are called macromolecules. Organic molecules also tend to be polymers. That is, while they're very large molecules, they're made up of repeating subunits, and the subunits are known as monomers. The other type of chemistry is inorganic chemistry. Now, inorganic molecules can contain carbon or they can contain hydrogen, but they cannot contain both. Typically, inorganic molecules are held together by ionic bonds. There are a couple of exceptions. Water is an inorganic molecule, as is carbon dioxide, and they are both held together with covalent bonds. Ionic molecules tend to be very small. These are acids, bases, salts, and water. These two types of chemistry are involved in biochemistry. Biochemistry is all of the inorganic and organic chemical reactions that are found in living things. Let's look at a few inorganic molecules first, and we'll start with water. Water is the largest component of living things. Water is very, very important to living things because it has some unique properties. Water has a very high heat of vaporization. This means it takes a lot of heat to vaporize water, to turn water into steam. Water also maintains its temperature very well. If you have a swimming pool or if you go out to the beach, you know that the air will be very cold, but the water will still be warm. Or in the springtime, the water will be very cold, but the air will start to warm up. It takes water a while to heat up, but once it's warm, it takes it a while to cool off. Water can be considered the closest thing we have to a universal solvent. It will dissolve most molecules. And remember that ionic compounds dissociate in water and become electrolytes. Because so many things dissolve in water, it makes water a good way to transport materials around the body. Water also can act as a cushion, sort of like a water bed. Many body cavities contain fluids that help cushion the organs. Also, water acts as a lubricant. The inorganic molecules, acids, bases, and salts have their basic characteristics because of how they dissociate. Salts will dissociate into cations and anions. So sodium chloride dissociates into sodium, which is a cation, and chloride, which is an anion. These will conduct electricity, so these are the electrolytes. Calcium salts help strengthen our bone, and iron salts help transport oxygen in our body. Acids also dissociate. They're held together by ionic bonds. They dissociate into hydrogen ions and anions. In other words, the cation they dissociate into is hydrogen. So hydrochloric acid dissociates into hydrogen ions and chloride ions. And bases dissociate into cations and hydroxyl ions. A hydroxyl ion is an oxygen and a hydrogen held together by a chemical bond, but it still has a negative charge. Sodium hydroxide dissociates into sodium and hydroxyl ions. Now, acids and bases change the pH of solutions. Buffers are chemicals that help resist pH changes. So exactly what is pH? Well, pH is the negative logarithm of the hydrogen ion concentration of a solution. That's really more information than you wanted, I'm sure. What you need to know about pH is that it is a scale that runs from 0 to 14. 7 in the middle is neutral. As you move from 7 toward 0, you're becoming more acidic, and as you move from 7 toward 14, you're becoming more basic or alkaline. The closer to 7 you are with pH, the weaker you are. The further from 7 you are, the stronger you are. So something with a pH of 6 would be a relatively weak acid. Something with a pH of 13 would be a strong base. Now here are some common things in their pH. Notice that pure water is neutral. It has a pH of 7. Seawater has a pH of 8. It's slightly alkaline. Black coffee has a pH of 5. It's kind of a weak acid, but lemon juice is a pretty strong acid. Oven cleaner is a pretty strong base. Now, before we had pH paper, the alchemists used to determine pH by taste. Things that are acid taste sour, and things that are basic are slippery and taste bitter. There are 4 organic molecules or macromolecules found in living things. Carbohydrates are our primary fuel. This is our primary energy source. Lipids are used for fuel storage. This is long-term energy reserves. Proteins are involved in building structures as well as doing certain functions in the cell. And nucleic acids store, retrieve, and transmit genetic information. Carbohydrates are made of 3 atoms, carbon, hydrogen, and oxygen. The building block or the monomer of a carbohydrate is a monosaccharide, something called a simple sugar. The polymer is when we have many of these monomers strung together. If we stick 2 monosaccharides together, we have a disaccharide. As we stick more and more monosaccharides together, we have a polysaccharide. This is a complex sugar or a complex carbohydrate. Carbohydrates function as our primary fuel and energy source. Now, carbohydrates can build structures. Cellulose is in the cell wall of plants, and it's what makes celery crunchy. And chitin is in the exoskeleton of insects. This is what makes the roach crunchy when you step on it. Our nutritional carbohydrates, the ones we typically think of when we talk about carbohydrates, are starch and glycogen. Now, here you see some of the monosaccharides, glucose, fructose, galactose. Each place that there's one of these points, unless there's something else written in here like that, is a carbon. So these are all called 6-carbon or hexoses. Deoxyribose and ribose are also simple sugars, but they only have 5 carbons, so they're called pentoses. As we push these together, we get long chains of these monosaccharides, and that's what makes a polysaccharide. This one happens to be glycogen. Lipids have the same atoms as carbohydrates, carbon, hydrogen, and oxygen, but in different numbers and arranged differently. The basic building block of what we call a neutral fat is glycerol and 3 fatty acids. Fatty acids are real long chains of carbon and hydrogen. We have different kinds of lipids. We have triglycerides as one kind of polymer, phospholipids, and then cholesterol. Lipids typically function in long-term energy storage. This is where you put energy that you're not going to get to for a while. Also, fats will help insulate the body, and fat pads help protect areas of the body. Now, the triglyceride is the basic lipid. Here is the glycerol molecule, and the fatty acids hang off like this, 3 fatty acids. This is mostly what you ingest. This is what's mostly stored in your adipose tissue, or triglycerides. Phospholipids happen because we break off one of the fatty acid chains and we stick a phosphate on it, this phosphorus-containing group. Now, this makes this molecule very interesting. The triglyceride we saw is not soluble in water, but when we put this phosphate group on this molecule, this end of the molecule is soluble in water. This end is insoluble in water. A lot of times they draw this molecule with what they call the polar head. Polar will dissolve in water, and a nonpolar tail. Nonpolar will not dissolve in water. So here we have a molecule that one end can dissolve in water and the other end can't. This is an important molecule in our cell membrane. It allows us to make a membrane that will interact with the watery inside and outside and yet still act as a barrier between the two. Cholesterol is not a chain, but rather this rather complicated structure. Again, there are carbons at all of these points unless some other letter is written in there. And cholesterol is a very stable molecule. So this is interwoven in our cell membrane to help stabilize its shape and structure. And it's also a major component of steroid hormones. Proteins are composed of carbon, hydrogen, oxygen, and nitrogen, and a few of the amino acids have sulfur in them. The monomer, or the basic building block of proteins, is the amino acids, and there are 20 different kinds of amino acids. As we stick amino acids together, 2 would make a dipeptide. Then we would have many that would be a polypeptide. Usually when we have a really long chain, we go ahead and call it a protein. Proteins have many functions. They help build cellular structures, parts of the cytoskeleton. They're involved in cellular identity. They help cells identify each other. They also are involved in a number of cellular function. Enzymes act as catalysts, antibodies help protect us from disease. The function of the protein is dependent on its molecular structure. If we don't have the right structure, we cannot recognize other molecules. Proteins are recognition molecules, so they have very specific shapes. Proteins have 4 different structures that we have to worry about. The primary structure of a protein is the exact sequence of amino acids, exactly which of the 20 amino acids are strung together. If we change one amino acid in a chain, we can change the protein. Normal hemoglobin and hemoglobin S, the hemoglobin that's involved in sickle cell anemia, has exactly 1 amino acid different, and yet we have a very different molecule just because of that one substitution. As a result of this chain of amino acids, we will have some opportunities for interaction between the molecules. This secondary structure is the coiling up of the chain into either an alpha helix or the rearrangement of the chain into what's called a beta-pleated sheet. This is the result of covalent and hydrogen bonds that occur between nearby amino acids in the chain. Then in our tertiary structure, we have even more folding. More hydrogen bonds form as a result of different amino acids coming in contact with each other from the secondary folding, and we get a very specifically shaped molecule. And most proteins stop here. But there are a number of proteins in the body that have quaternary structure. That is, 2 or more polypeptide chains are linked together for some reason. Here are some examples of amino acids. Every amino acid will have this amino group and this carboxyl group, and these are the 2 reactive areas of the amino acid. The rest of the molecule, this side group, can vary, and this is what makes each of the 20 amino acids unique. Here, glycine just has a hydrogen as the side group, but phenylalanine has this cyclic compound as its side group. And cysteine is one that contains sulfur. It has a carbon and a sulfur molecule attached there. Our primary structure is sticking these things together. And they stick together at this nitrogen carbon carbon. Notice it's nitrogen carbon carbon. And when we stick them together like this, these side groups are sticking out. And now this side group is kind of close to this side group. And this may allow them to interact and make a hydrogen bond or a covalent bond, and this begins the secondary structure. So your primary structure, again, interactions with these hanging out groups here, and you either get this alpha helix or this beta pleated sheet, and you'll see hydrogen bonds between various bits and pieces of the different amino acids. That's what holds those in place. Then we have further folding. This is the tertiary structure. And we can have, in this tertiary structure, the entire protein may have areas of alpha helix and other areas of beta pleated sheet. So it's not like all of one protein chain is just one thing, either alpha helix or beta pleated sheet. But this allows them to fold up and give you a very specific shape. Now, if 2 or more are hooked together, then you have a quaternary structure. This could be hemoglobin. Iron would be the molecule in here that would be holding these 4 proteins together. Because protein shape is so important to the function, if you alter the shape, you're going to alter the function. Now, when you alter the shape of a protein, this is known as denaturation. Denaturation disrupts bonds, which disrupts the shape. Denaturation can be reversible, that is, if you stop the disrupting force, the protein may fold back up like it's supposed to be, or you may so severely disrupt the protein that it cannot refold, in which case it's irreversible. Denaturation can be caused by 4 things. One is pH. If you change the pH, you're going to change the protein shape. If you take a little bit of egg white is almost pure protein, and you put it in a little bit of vinegar and let it sit for a while, you'll see that the egg white turns white like it's been cooked. Now, temperature is another thing that denatures protein. You've all cooked eggs, and you know what the egg white looks like. You've changed its structure. Ionic concentration And mechanical agitation will also denature protein. And if you've ever made meringue, taken egg whites and beaten them, the mechanical agitation will begin to disrupt. And then if you add sugar or cream of tartar, you're changing the ionic concentration, and that will kind of stabilize the denaturation. Enzymes are functional proteins. Now you always know something is an enzyme because it ends in ASE. Enzymes are what we call biological catalysts. Catalysts in general speed up chemical reactions. They're not part of the reaction, they're unchanged by the reaction, but they make reactions occur much faster than they would normally. In living things, our catalysts are always enzymes, these proteins. Enzymes function by lowering the activation energy of a reaction. Activation energy is the energy that you've got to put into a reaction to get it started. Now you do this every morning when you start your car. Your starter creates sparks, and those sparks will ignite the gasoline so you can start those little explosions in your motor that move your pistons. If your starter doesn't work, gasoline doesn't spontaneously do that. So that spark acts almost like a catalyst. It lowers the activation energy of getting gasoline to burn. Now here you see an example of how enzymes work. Here is this hill, this activation energy, and we have to get to this point before the reaction will occur. What an enzyme does is it simply flattens out that hill a little bit, meaning the reaction can start faster. If it can start faster, then it will occur faster. Enzymes have to fit their substrate just like a puzzle or a lock and key. So enzymes have a specific shape. They fit only one substrate. They're specific for one substrate. When their substrate, or whatever it is they work on, that's what a substrate is, what they work on, fits to the enzyme, the chemical reaction occurs. The material leaves, the substrate leaves, and it's been changed. But the enzyme is free and it has not been changed at all. It's capable of entering into another chemical reaction. Nucleic acids are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. The monomer is the nucleotide. Now, a nucleotide is made of a pentose, a 5-carbon sugar, either ribose or deoxyribose, a phosphate molecule, and a nitrogen base. The polymers are either DNA or RNA. And as we will see later, these function to store, retrieve, and transmit hereditary information. Here you see a DNA molecule, its bases, its deoxyribose, its phosphate. And DNA exists as what we call a double helix. And here's an example of some hydrogen bonds that are holding molecules together. RNA, here we have ribose and phosphate, and we've got some different bases. It tends to be a single strand. Adenosine triphosphate, or ATP, is a very special nucleotide. This is the nucleotide that stores energy in chemical bonds. This energy is the source of immediate usable energy in the cell. Adenosine triphosphate is an RNA nucleotide. It has adenine and 3 phosphates, so it's ribose, adenine, and 3 phosphates. Just putting together an adenine, a phosphate, and ribose doesn't take a whole lot of energy. To add the second phosphate takes a little additional energy, but to add the third phosphate takes enormous energy, and that energy is stored in that chemical bond. ATP is how your cell manages energy. If you eat a Snickers bar, there is a tremendous amount of energy released all at one time in your cells. That's more than your cells could take. They would explode. So we quickly store a lot of that energy in ATP. That ATP can sit around, and 5 minutes later, 3 minutes later, whenever you need a little energy, we can break down the ATP and get the energy back out. Now here you see your basic nucleotide, adenine, your ribose, and 1 phosphate. The little wavy line here indicates that this is an energy bond. Straight line means not too much energy was used here, but this is a high energy bond. When we put that second phosphate on, we have ADP. And when we put the third phosphate on, notice there's another little wavy line that gives us ATP. Now in the cell, when we remove those phosphates, we're going to release energy. And this is high energy storage. Back and forth this goes. ADP plus energy plus phosphate will yield ATP, and if we need the energy back, we break down the ATP, we get the phosphate back, we get the energy, and we get ADP. And we can go back and forth based on whether we have extra energy we need to store or whether we need to release energy. ATP in the cell is used to move material across the membrane to transport stuff. It's used for mechanical work like muscle contraction, and it's used for chemical work. Whenever we put two things together, it takes some energy to make that bond, and we can use ATP to help make that bond.